Sounds great. So before I get started, I do want to just, of course, remind you that there's a couple of really good resources out there available because, this is kind of a short presentation today. You may have other questions. Of course, my website, badgercropdoc.com, and then also the Crop Protection Network site where you can find almost all things pest management now on that website. Also, my fungicide test summaries are a great resource. Those are out. Those came out last fall and are available, or last spring and are available at that QR code there. Been getting a lot of questions, on fungicide performance and efficacy and some of those things. And I would say, this is a good resource. You're probably really interested in 2023, but don't forget to go back. There's over a decade of these reports now, and you kind of want to go back and look at what's happened over time, as well as across multiple locations, and choose products that have efficacy that are consistent across those locations and time. Now, really our focus at this point in the season has been on winter wheat. Winter wheat is moving very quickly. We do have a couple of disease concerns this year. The last couple of seasons, we haven't had much of a disease issue because it's been generally hot and dry in the spring. This has changed this year. We're cooler and wetter with some heat starting to come in now as we get these more frequent thunderstorms. The two main diseases of concern at this point are going to be fusarium head blight or scab, and then also stripe rust. And you probably heard me talk about these before, but I do want to just give a little refresher in where we're at here. Remember that, stripe rust is actually part of a complex of rust. So you have leaf rust, stripe rust, and stem rust that all affect wheat. But due to good genetic resistance, we generally don't see much stem rust, and leaf rust is generally only on highly susceptible lines. So that leaves us with stripe rust, and stripe rust is actually the coolest loving rust of the three. So what that means is that it likes cooler temperatures, kind of like what we're seeing now. So if we have rain, cool temps, it can move into the area and affect wheat, and especially if it moves in around that flag leaf emergence timing or afterwards, it can affect yield up to a point. Just to show you the stripe rust cycle, this rust can overwinter in some years. I had a student, Brian Mueller, a few years back. He looked at the ability of this particular rust to be able to actually overwinter in Wisconsin. And in years where we have good snowfall or snow depth over the winter, and we have an early epidemic in the fall, it can overwinter in those years. I would estimate it's 1 or 2 in 10 years where that actually could happen. Given the fact that we haven't had really any stripe rust epidemics over the last 2 seasons, we really don't have any residual inoculum here in this state. And we didn't have much snow cover through the winter this past year. So really we haven't seen any overwintering rust in a number of years now. So what that means is that we call these Uridinia spores, they actually have to come up from the south. So the Louisiana kind of Gulf Coast area, that's where they overwinter, and those have to be brought up on weather systems. So typically we watch what's going on what we call the rust pathway. Once it moves up the rust pathway, gets into Wisconsin, it can infect a few plants, and then we get these repeating cycles as those pustules actually serve as the source of secondary inoculum. And if that ramps up really quick, we can have a really fast epidemic progression. And the faster that epidemic progresses, the more yield loss we can actually have. Contrast this with fusarium head blight, which is a little different fungus here. This is what we would consider a soil-borne fungus. So it's hiding out on corn residue, wheat residue that's in the soil. And then when we have wet, rainy conditions, we get those spores actually rain splashed up onto the anthers of the wheat heads. So because you have to have those anthers out, that's the window of opportunity for the fungus. So before the anthers are out, there's really no risk for infection. And then after the anthers go away, there's really no risk for infection. So the window of opportunity for the fungus is pretty narrow. But if it happens to infect, it can colonize down those kernels and cause shrivel kernels. And then we can also get secondary metabolites produced like mycotoxins. Specifically deoxynivalenol or DON or vomitoxin is the big one. So not only do we get yield loss, quality issues with the seed, but we can have high vomitoxin loads as well with this particular disease. Now, how do you sort of track these two diseases? I mentioned first with stripe rust, we have the rust pathway. So we in Extension, the National Extension Network, actually follow the stripe rust over the season. We actually chart this on a national map. You can visit these maps there at that QR code. And so what we watch is what's going on down here in Louisiana, and then what's coming up the Mississippi River Valley. And you can see this, I did a screen capture of this map a couple days ago, putting this presentation together. And you can see it has made its way up as far as Michigan. And what we like to watch for is once it gets into Illinois, we start to do some scouting in Wisconsin. And I did send out an article or a blog post a couple of weeks ago where we urge people to get out and start scouting because we saw that Illinois county light up. Now, the wheat crop is moving very quickly phenologically, so we're already to heading. I looked at my own research plots today. We're going to have anthers out probably on Friday. We'll actually be doing our scab applications hopefully on Friday if all goes well. And so we're about 2 weeks ahead of where we typically are this time of year. So where I'm going with this is even if stripe rust moves in at this point, especially into southern, south central Wisconsin, I think we've escaped the heavy yield hit that we might get from this particular disease on most varieties. So there is some good news here, but I would urge you to continue to keep track of this and watching what's going on as the season progresses. When it comes to head blight, we have another system that we use, wheatscab.psu.edu. If you scan that QR code, it'll take you to the site. This is a screen capture from a couple of days ago, looking at the head scab risk. Right now on a susceptible variety is showing low risk. This is a little suspect for me. We've got enough rain here. I'm expecting this to turn around pretty quickly if we can get some of this heat that we're seeing in the forecast. So what I'm thinking at this point, we probably need to put that fungicide application out for head scab, and we're going to also be able to control stripe rust with that fungicide application here. And so we're going to talk a little bit about how do we make that decision in terms of that when to go with that fungicide application timing. In terms of the actual products, we have a couple of resources. Probably the best resource is the Crop Protection Network resource, which is the fungicide efficacy table for wheat diseases. This actually just came out about a week ago, the newest update, and you'll find all the major wheat diseases on here and efficacy ratings. I contribute to this, so there's Wisconsin data in here, but it is a national effort. So you're going to get to see, really what the consensus is among these products nationally against the various diseases. And if you look at that table, there's about 4, maybe 5 products really that have a good efficacy rating against head scab, and those are the ones you're going to want to focus on. Fortunately, if you're trying to kill 2 birds with 1 stone and you're trying to take care of the stripe rust situation at the same time, all the products rated good for head scab are usually rated excellent or at least good or very good against stripe rust. So you're going to be able to take care of both diseases with a good, well-timed head scab application. So that brings me to how do we make this head scab application decision? It's really all about anthesis. I'm showing you the 2 kind of major times that we want to apply a fungicide. The first would be Feekes 8. We're generally in southern and south central Wisconsin. We're probably already a bit past that Feekes 8 or emerged flagleaf. That would be a good kind of stripe rust timing if we saw stripe rust moving in. But what we really want to focus on now is this 10.5 and specifically 10.5.1 timing where we have heads out, and more importantly, where we're starting to see the actual anthers. So in this early timing here, you can see this is the 10.5.1. So if you have 50% of the plants approximately in the field showing at least 1 anther out, like you see in this left-hand photo here, then that would be the 10.5.1 timing for that field. Okay, now what we've learned over the years is that we can actually delay those applications a bit, and we do see a lot of unevenness in some years in terms of head emergence, which complicates this decision a bit. The good news is that you can sort of start at 10.5.1, but really have about 7 days after that start time to make your fungicide application. And in some of our data, and I've showed this here in the past, we actually have seen better control of vomitoxin actually, where we delay those applications 5 to 7 days after the start of anthesis. So you do have a window there. That window is short, so if it gets wet, that is complicating, but at least we do have some flexibility in terms of that application. I've showed this data before. I'll just remind you that this is out there, and I would encourage you to go back and look at this. This was some work that we did with a postdoc a few years ago, Sean and I did, where we looked at sort of just the regular old management that we would do with wheat, typical here in Wisconsin, where we might ramp that up to what we call a mid-level and then high level. And really the difference between the mid-level and high level are some micronutrient packages, as well as a 1 versus 2 fungicide application strategy. So in the years where we did this, 2016 and 2017 happened to be heavy stripe rust years. So in those years, 2 fungicide applications were really beneficial. But in the other 2 years, 2018 and 2019, those were primarily head scab years. So we just really actually needed the second application of that 10.5.1. We're already kind of past the flag leaf application timing at this point. So we really want to focus on that 10.5.1. And that's what we would term kind of the mid-level strategy here. And you can see even with the mid-level, we get really good control of DON with that application. That's really the key there is you want to kind of reduce those test weight deductions as well as the DON deductions. So you want to place a lot of focus at that 10.5.1 timing. And like I mentioned, you're going to pick up the stripe rust control anyway if it happens to move in. And you can kind of see it here in this data. We took a look at it from an economic standpoint. We're really preserving, having no deductions essentially, and really good quality wheat with that 1 fungicide application around that anthesis timing. So there's a lot of value in that application. There's a lot of value in getting that application timing correct. We even took this a little bit further, looked at the ROI there, and you can see the mid-level actually edged out the high level in terms of maximum ROI. All the programs gave us a positive return on investment, but you can see it was a really substantial increase over the current where we applied that single fungicide application. So again, I think in 2024, where we stand at this point, I would put that application of fungicide on here as we get to this anthesis timing, or target it at least maybe 5 to 7 days after the start of anthesis, and don't even look back. Here's the take home generally around the timing thing. I would plan to apply that fungicide, or that fusarium head blight application now. Have that on your radar. I know some fungicides have already gone out in far southern Wisconsin. I didn't talk much about going a little earlier than anthesis. I don't generally recommend that. That's a little bit too early in my opinion. But if you do look at like the Miravis ACE label, which is one of those few products that is rated good, you can actually go a little bit earlier according to that label. The issue there is that we don't see as good at vomitoxin control at that earlier application. It is better than not doing anything at all, but you're not going to get as good vomitoxin control as you might if you waited for that anthesis or 5 to 7 day after anthesis timing. So place your focus there, get that timing right, choose a good product that's indicated on that label. All right, I want to just shift gears to a little different disease now on soybeans that we've been kind of following, and this has got our attention a bit this year because we are, a lot of soybeans went in the ground. Now we've gotten wet, and so we're sort of expecting, and we've actually seen some emergence issues in our own research at this point. And we're expecting some what I call oomycete root and stem rot problems. And what are the oomycetes? These are the water molds. Phytophthora, you probably know, Pythium, and Phytopythium. So these are our seedling and seedling and stem rot issues that we can see early in the season after planting, especially when it's wet. This is some work that Sarah D'Souza, who's a master's student in my lab, has been conducting. We've been trying to take a look at the oomycete population in the state, specifically for soybeans, and how that is playing out here in the state. Just to briefly touch on the life cycle, generally, these three genera of water molds, they overwinter as what we call oospores. So these are hardened spores that can survive multiple years, generally on crop residue. And what's interesting about these is that when we have, saturated soils, 60-degree or so temperatures, these oospores can actually form what are called sporangia, which give rise to motile zoospores. So these are spores that can actually swim in water films in the soil. So when you have standing water in a field or you have waterlogged soils, these zoospores are germinating, they're finding roots, and they're infecting the roots and working their way into these seedlings and causing seed issues, seedling blights, and all those sorts of things that we see with stand loss or associated with stand loss, especially in wet conditions. So again, just to touch on the favorable conditions here, saturated soils, poorly drained soils, compacted areas of the field, and warmer temperatures, although I'll talk about pythium here in a minute. There's really a pythium for every temperature. So I don't get as worried about the whole temperature thing, but it's really all about how much saturation we have in some of these heavier soils, especially. Just to show you some of the symptoms that can be caused here, we can have root rots, we can have hits on the lower stem portions in these newly emerged seedlings. So you want to be looking and digging and washing roots and kind of taking a look at what's going on there. You can see the roots basically blasted off these seedlings here. So obviously those seedlings are going to eventually die because they don't have a root system that can actually sustain the plant. So do a little bit of scouting, a little bit of homework, dig some plants if you're seeing some of these above ground symptoms, and make sure you know what's going on below ground. You can also send samples into our diagnostic clinic and we can help there as well. I want to talk just a little bit about the work that Sarah's been doing in the state to try to get a handle on this. This is ongoing research. This is the summarized data from the 2022 season. So this is a long project in that we get soil samples, say in 2022. It takes us the winter of 22 and 23 to actually process those samples. And then we got samples again in 2023, and we're in the process of actually processing those samples. So we're almost a year behind in this, but what we're doing is we're actually surveying the state to look at the population of these oomycetes in areas where we grow soybeans. And you can see where we had collected in 2022, the more intense the red in these particular counties, the higher the number of samples. We did sample in 39 counties in 2022, and then we went back and tried to fill in counties in 2023 where we didn't have samples. So we're hopeful after she's done processing all the 2023 samples that we'll have actually looked at soils from across the state. So what we did is we worked with Pest Pros. These are samples that come in through the soybean cyst nematode program. Pest Pros was generous enough to give us those soil samples after they took their soil out for the soybean cyst nematode program. We brought those down to campus. We air dry those samples, and then we do a styrofoam cup assay where we actually flood the soils, dry them again, flood them again. That gets those zoospores to actually swim. And then we can do this soybean leaf disc incubation where we can actually trap those zoospores on those leaf discs, and then use a selective media process where we actually place those leaf discs out, and then you can see the organisms actually growing out on the surface here. So this is a labor-intensive project, takes a long time to process those samples, but we're starting to learn a lot about the oomycetes that are actually colonizing some of the soils here in Wisconsin. Once we have the organisms growing out and they're in pure culture, we're actually using DNA techniques to actually look at what those species actually are. So we're not relying on morphology. We're going right to the DNA. We extract that genetic material. We amplify it through a process in the lab. Then we can actually do what's called sequencing to actually get that genetic code. So we're sure we actually know what those species really are. So just generally in Wisconsin, what have we found? We were expecting to actually see high levels of phytophthora. So that was interesting. And what we're actually seeing is actually higher levels of pythium problems. And so we generally get a lot of pythium. We pick up a few phytopteras. You can see the frequencies there. But of course, pythium is the one that really seems to be the thing that we're getting the majority of in terms of positives. And what's interesting about Pythium is that it is also a pathogen, or at least there's multiple species of Pythium that are also pathogens of corn as well. And so where we have soybeans in rotation with corn, we expect that some of that is contributing to the high level of Pythium that we actually get here in the state. And you can see the distribution. Triangles are Phytophthora, the pluses are the Phytopythium, and then the circles are Pythium. And obviously you can see Pythium across much of the arable acreage that we have here in Wisconsin, where we grow a lot of soybeans and also corn. So that really does seem to be the genera that are really comprise a lot of the population here in Wisconsin. Just to break it down more specifically to actual species, now you can see Pythium irregulare, which is we picked this one up a lot. Pythium torlosum, Pythium inflatum. These are all known pathogens of soybeans here in the state, and you can see their frequencies are pretty high. And then way down, kind of mid-level here, we get the two Phytoptheras, which are typically a problem for us, Phytophthora soji, and then the lesser-known Phytophthora sansomiana. So they're actually less frequently occurring than we originally thought. And this is in line with some other work in other states like Ohio, where they've looked at this as well. They also pick up a lot of Pythium, and it usually is at much higher frequency than Phytophthora, in fact. So what does that mean for us here in Wisconsin? Well, the frequency of Pythium is high, much higher than Phytophthora. I think we got to really start looking at Pythium more intently. We need to learn more about this particular group of organisms. It is a problem on corn as well, so we might even need to be looking at our rotations and maybe taking a hard look at the seed treatments that we're actually going to be looking at as well. We need to actively manage early season Pythium problems on soybeans and corn, especially in a year like 2024. Keep your eyes out, looking at these things, be out there scouting, targeting fields that you know have heavier soils, poorly drained. Those are going to be the areas where you might run into these problems. In terms of management, when it comes to Phytophthora, we do have those RPS resistance genes. So if you're seeing what you think is Phytophthora, you get a confirmation of a Phytophthora problem from a diagnostic clinic, go back to that variety and look up what RPS gene is there. We're also interested in RPS genes and resistance here and what might be overcoming certain levels of resistance. So we're certainly interested in that. When it comes to Phytophthora sansomiana and the Pythiums and Phytopythiums, however, we have really no genetic resistance against those particular organisms. So we're relying more on, say, just general resistance that might be there. We place a lot of, we're placing a lot on our seed treatments right now. So mefenoxam, metalaxyl, ethoboxam, oxyproline, you can never say that one. And, of course, we have some cultural practices like delaying planting time, although we don't generally want to do that with soybeans. We want to get in there and protect that yield potential, but that certainly is a strategy. And then, of course, improving drainage as well can obviously help with these oomycete pathogens. I mentioned the seed treatments. I've been getting a lot of questions there, and especially if you see problems, make sure you know what the seed treatment was. Not that you can do anything now on this particular crop, but you can do some things to try to go back and see what seed treatment was on there. Did it work? Did it not work? Understanding what's actually there. We have a couple of resources for that. So the fungicide efficacy tables also exist for soybean seedling diseases. So these are all seed treatments. There's a QR code there on the left. And then on the right, we have our own What's on Your Seed publication. It's from UW Extension here as well. And you can find that particular document at the QR code on the right. That was just updated this past winter. So it should be relatively up to date. And what that does is that tells you generally what the trade name of products are, and then what the actual active ingredients are in those seed treatments and what they're actually targeting. So it can help you sort of wrap your head around what you're actually applying out there on those seeds. So with that, I know I was really fast, but hopefully I left a little bit of time. I could answer some questions. I certainly need to acknowledge my team here. You can see the various graduate students, technicians, and folks who help us get this all done there. You see my contact information there, Badger Crop Doc on Twitter or X, whatever we call it these days. And of course, email is an excellent way to get in touch with me. So with that, I will stop share and I'll turn it back over to Sam and company.